Hello guys, welcome back. We are going to be going over carbohydrates, so let's begin. So here you can see I've uploaded the specification. You can get this off the AQA website and use the specification as a guidance throughout your journey through A-levels, as I've mentioned this before. So I'll be going through each and every section of the specification to ensure that you've got sufficient information to take into the exam. So let's begin. So let's look at the first point. We are looking at this. So monosaccharides are the monomers from which large carbohydrates are made. So let's break this down a little. So we talked about monomers, or you should know what monomers are before you even look at this lesson. So monomers are small repeating units which chemically bonded together make a polymer. Now we have different types of monomers, and one we've just been introduced to is monosaccharides. Now examples of these monosaccharides or these monomers would be glucose, galactose, and the last one is fructose. So don't worry, I'll be going through each and every one of these. So like I told you about the analogy of the Lego brick, so this one piece Lego brick is a monomer. The same with having, let's say, a glucose molecule. So pretend this is a very simplified glucose molecule. We have one glucose molecule. This is a monosaccharide. Join these Lego bricks together, join these Lego bricks together and we get a chain. So joined by, let's say, a bond. Well, in reality there isn't a bond between Lego bricks, but there is, just imagine one. So we get a chain of Lego bricks. Same here, if we have lots of glucose molecules, just going to quickly draw this. If we have lots of glucose molecules, join them together and we get something called a polysaccharide. Now you should know what a polysaccharide is, even though you haven't studied it or know what it is, because you've got this prefix here, poly, and poly means many. And like I said before, possibly I don't think I have said it, but I'll say it again, is every time you see the word saccharide, assume carbohydrates or sugars. So let's look at these terms here. So I've already mentioned what a monosaccharide is. It's basically a monomer, which is a carbohydrate. Now we've got this thing here called disaccharides. Now you should know, even without knowing that it's a monomer plus another monomer, you should know immediately that it's involving two monosaccharides because it's got the prefix di in it, and di means two. So we've got two monomers or monosaccharides which are joined together. Then we've got polysaccharide, like I mentioned before, and this is more than two monosaccharides chemically bonded together. Now all carbohydrate elements that you'll come across during A-levels will contain the elements carbon, hydrogen and oxygen. Monosaccharides are the monomers which make up carbohydrates, so they are the building blocks. Don't use the term building blocks in the exam, but just as for ease and of understanding, these are the building blocks of carbohydrates. Glucose is a monomer, like I said before, and it's on the specification, and this glucose is also referred to as a hexose sugar. This is because it's got a hexagon shape when we draw it out. And there are two isotopes. So isotopes is basically two different forms. You don't need to go into chemistry of this, but it's just all you need to know there are two different types of glucose molecules or monomers that you need to know about, and you need to know about their structure. So here are the two monomers, glucose monomers, that you need to know about, and these are the exact diagrams you need to copy, and they're copy and pasted from directly from the specification. So don't go bother go around and looking to other textbooks and drawing their diagram. Just draw exactly how the AQA specification has done it right here. And the only differences between these two is the right hand side of the two monomers, and you can tell straight away that they've just been inverted or flipped. So you need to find a way of memorizing these diagrams without getting confused. So the way I used to do it, or still do, is because this glucose here is called alpha glucose, A for alpha, and then you've got H, and then a hydroxyl group, or you can call this an OH group. I just put HOH, so I know that's first, that goes on the top, and then I've got my hydroxyl group. So you can pronounce this AHO, or whatever you want to do. You can make up a mnemonic, do whatever you want, find the best way to memorize this. And then for beta glucose, you've got B for beta, and then you've got the first molecule here is OH, then we've got a hydrogen. So just remember, this is called a hydroxyl group. So we've got B for beta, and then the rest of it just makes the word O. So you can call it BO or BO or whatever you want to say. And just practice drawing these diagrams out. And they are usually around about 2 markers in the exam, so quite precious knowing how easy this actually is. Here is another diagram. So you might want to draw this if you find this diagram a lot harder, but I prefer you to use blah blah blah, sorry about that. So I prefer you to use this diagram only because the AQA specification exam board have provided this diagram. But you can get away with using this diagram as well, and the benefit of this diagram is that it's a lot more clearer and you can see where the bonds are, how many carbons there are. So when I mentioned that glucose is a hexose sugar, hex basically means 6, and every time you see the word, the suffix ose, think sugars. Here you can see that it's got 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbons, 6 carbons. It's got 6 carbons in total, and the hex is referring to the number of carbons. Same for beta glucose, same for alpha glucose. Okay, so you need to be familiar with the other 2 monomers. So we've had a look at glucose and the alpha and beta glucose. So you also need to be familiar with the structure of galactose and fructose. Yet they haven't necessarily tested this in the exam before, but you've also got to be aware of their structure and differences. So don't take a chance. So just if you want those extra marks, or if you want to be in the safe zone, I'd recommend you memorizing these 2 diagrams alongside obviously memorizing the compulsory glucose monomer. So going on to the next part of the specification on carbohydrates, we've got these things called disaccharides. Like I mentioned before, you've got 2 monomers or monosaccharides chemically bonded together. Now disaccharides are formed by condensation, and you should remember what condensation is because you'll be using it a lot. And condensation is basically adding 2 molecules together, forming a chemical bond with the release of a water molecule. So you need to know a few specific disaccharides and how they are formed. By how they are formed, I mean what are they made from initially, so the monosaccharides that make up these disaccharides. So let's look at the first one. So maltose is a disaccharide. So it's a molecule with a compound with 2 monosaccharides chemically bonded together. And this is formed obviously by the process of condensation, and it's made by 2 alpha glucose molecules. So glucose plus glucose makes maltose, plus obviously your water molecule that has been last eliminated. Then you've also got sucrose. Now sucrose is also another disaccharide. It's formed by condensation, so it's joining two molecules together, monomers together, to form a chemical bond and the release of water. So here we are going to have glucose as the first monomer, and the other monomer is fructose. So glucose plus fructose makes sucrose. Notice how each of these end in ose. Like I said before, every time you see the word ose, or sorry, the suffix ose, assume immediately it's going to be a sugar. So they might hit you up in the exam with something like tetradiamine hexamine ose. Every time you see the word ose, don't be put off, you know it's a sugar and you're referring to the topic carbohydrates. So so we've got glucose plus fructose makes sucrose plus water. And the last one is lactose. So lactose is a disaccharide formed by condensation, like I mentioned before, and of a glucose molecule and a galactose molecule. So glucose plus galactose makes lactose. Now the best way to remember this, well, firstly this one, so let's label this 2, 3. So equation number 2 is quite easy to memorize, because you've got galactose, which is a monosaccharide, and within galactose you have the word lactose, and lactose is a disaccharide. So this one is relatively easy to memorize. Glucose plus glucose makes maltose. You've just got to know that. I can't think of any way to memorize that unless you want to play around with mnemonics. You can do G, G, M. You can, for this one you can also do G, G, L, and this one you can also do G, F, S. And then another one for the last equation is, to be honest, there isn't one. I can't think of one. But if you can think of one, then comment it down below. So disaccharide is formed by a condensation reaction, like we mentioned before, where two monosaccharides chemically bond together, releasing a water molecule. So a hydroxyl group, so you probably remember me mentioning that every time you see the word OH, assume that they're going to call this a hydroxyl group or an alcohol group or an OH group, but refer to it as a hydroxyl group. So a hydroxyl group from one monosaccharide and a hydrogen from another are removed to make a water molecule, obviously because you've got OH plus H makes water. So we've got hydrogen from one monosaccharide and hydroxyl group from another monosaccharide, and this makes water, and as a result we also get a chemical bond. So the specific chemical bond we make is called a glycosidic bond, and the main feature of this the formation of this glycosidic bond is that it increases the carbohydrate chain length. This might be a bit obvious to just say, but it's directly from the mark scheme, so I'd mention this or write it down in your notes. Knowing what I've mentioned so far, let's see if you can attempt this. So it's relatively kind of hard doing it first time round, but it's good that if you try. So we've got two alpha glucose molecules, so we know that alpha plus alpha, or alpha glucose plus alpha glucose, makes maltose plus your water. Now we know the structure of alpha glucose, and we know what happens when these two chemically bond together. Obviously we're going to release a water molecule. So like I said before, we are going to remove a hydroxyl group from one monosaccharide and a hydrogen from another, like this, and then we get the release of water, and as a result we form this glycosidic bond here, and you can see here that this oxygen here is left out and as a result attaches to the other monosaccharide and forms what's called a glycosidic bond. And you need to know this this phrase term glycosidic bond, and it's specific to only carbohydrates. Here we've got this kind of overwhelming equation. So the definition of a carbohydrate is basically a sugar polymer. Now carbohydrates are made up of carbon and water. So I mentioned before how all carbohydrates contain the elements carbon, hydrogen and oxygen. And don't get confused by this, that they're made up of water as well, because water is literally H2O and we've got hydrogen and we've got oxygen. So the main elements that are within carbohydrates are carbon, hydrogen and oxygen. And as a result, some genius scientists came up with an equation which can give us the formula of glucose or any other carbohydrate. So the equation is CnH2ON. Now, N is the number of carbons there are, and then we've got water, and then we've got N being the number of water molecules there are, or N is just the number of carbon atoms. That's it. Don't confuse yourself. Just N is the number of carbon atoms. So going back to this equation that we visited earlier, do you think you can form an equation for glucose? And can you form the general, the general formula of glucose and general formula of fructose and maltose? So here we have maltose, and here we have glucose. But can you also draw the general, write down the general formula of fructose from your notes, and using this equation right here? So pause the video and have a go. So because we know that alpha glucose is a hexose sugar, and because we know that hex means 6 is referring to the number of carbon atoms, therefore N is going to be 6. Basic maths. Okay, so 6, so we're going to put C6 following this equation right here, H. Now as you can probably notice that 6, so we replaced the N with the 6. Now you would never ever write it like this in the exam, so you would kind of like have to expand the brackets. So you would do 6 times 2, which is 12, so C6 H12, and then obviously we don't write 1, but there's a 1 right there because there's 1 oxygen, and we do 1 times 6, which is 6, so we get C6 H12O6, which is the general formula. Now to write the general formula of maltose might be slightly harder, or it might trip up some people. Well, we know that it's made up of 2 glucose molecules, so we've got the general formula of glucose right here, so it's C6 H12O6, and we're just going to times that by 2. So obviously 6 times 2 is 12, 12 times 2 is 24, and 6 times 2 is 12. So that should be our general formula of maltose. But you're wrong. If you put that down as the general formula, you are incorrect. The reason being is because you've forgotten that this is a condensation reaction, and as a result of that we lose a molecule of water. So we have to subtract a water molecule from this equation right here, general formula. So all we have to do is knock off 2 hydrogens, so that gives us 22, and knock off 1 oxygen, which gives us 11. So we get C12 H22O11, which is the general formula of maltose. And if you really want to, you can check that out and count it all up. Now fructose, some people might be like, wait, what's fructose? Well, fructose is just slightly different. We know fructose is not a hexose sugar, but in fact a pentose sugar. Now please do tell me you know what pentose means. Pentose, obviously ose means the sugar, pent obviously is a prefix for 5, so it's referring to the number of carbon atoms. And so that's it, that's literally it. So we just substitute 5 into the formula, so C5, and then we've got H2O5. Now we're going to expand the bracket, so it's going to be C5 H10O5, and that's the general formula for fructose. Now let's have a look at polysaccharides. So polysaccharides are formed by the condensation of many glucose. So polysaccharides are 2 or more monosaccharides joined together. Let's take a look at 2 examples that you need to know about in the exam. 2 polysaccharides that you need to know about: glycogen and starch. Starch you find in plants, glycogen you find in humans or animals. Now starch is made up of 2 polysaccharides, amylose and amylopectin. Now don't be overwhelmed, it's literally, we know that starch is a polysaccharide, so it's a long chain of monosaccharides. The only difference is that we've got another chain. One's called amylose and one's called amylopectin. That's it. Don't overconfuse yourself. We've got 2 polysaccharides right here, and that collectively makes up starch. So it's a chain of alpha glucose molecules, and it makes a polysaccharide called amylose. So when starch we know consists of 2 polysaccharides, amylose and amylopectin. So amylose, the way we make amylose is by having adjacent alpha glucose molecules, and collectively that makes up amylose. Looks something like this. So we've got 1 alpha glucose here, another one here, another one here, another one here. Hopefully you should remember what this bond is right here, what it's called. This is called a glycosidic bond. But to be more specific and to get that A star, we are going to call it a 1,4 glycosidic bond. Now don't get confused as to where the 1,4 came from. Let me just show you where it came from. Reverting back to this diagram right here, we've got carbons which are labeled 1, 2, 3, 4, 5, 6. It's labeled from far right to left. Same here, far right to left. Now when 2 alpha glucose molecules decide to form a bond, they come across adjacent to each other, and as a result we have carbon number 1 right here is adjacent to carbon number 4 on the other monomer or monosaccharide, and these 2 carbons end up forming a bond next to each other, and as a result, because this is called a glycosidic bond, and because it's carbon number 1 and carbon number 4 which are forming the bond, we call this 1,4 glycosidic bond. So via condensation reactions of many glucose monomers, the important word here is many, making water and a polymer which are joined by glycosidic bonds, specifically 1,4 glycosidic bonds. Don't forget, for every bond or glycosidic bond we make, we release 1 water molecule. So it could possibly hit you up in the exam with a question. So the AQA could come up with a question saying, here is a 7, so they might show you a diagram here, and it will say how many water molecules were lost. Well, then you can say, well, there's how many glycosidic bonds are there, so ignoring these end bits, we can say there's 1, 2, 3. So there's 3 water molecules that were lost. So carrying on with starch, so we've just had a look at amylose, and this is what amylose looks like. Now, the structure of starch in relation to its like function. So number one, it's insoluble, so it can't mix with water, so it doesn't affect water potential. So water potential is basically saying the concentration of water, if I had to put it in very simple terms, so how much water there is. Number two, it's branched, and so it's coiled, so like you can see here, there's lots of coiling going on, and as a result it makes it quite compact and can fit into one area. Now you can imagine like loose string, etc., and if you scrunch it all up into like a bundle, it takes up quite a lot of space. But then if you wrap it around neatly around, let's say, a tube, it will take up less space. So because it's a polymer of alpha glucose, it provides energy for respiration, and as you should know from GCSEs, glucose is used in respiration to make energy. Now because we've got amylopectin, which is branched, as you can see here, that means there's more surface area, and therefore enzymes can act upon it a lot quicker to break it down faster. Now because it's relatively quite large starch, it can't leave the cell surface membrane. So sorry, here I've made a mistake. You must mention cell surface membrane. So not just cell membrane, but cell surface. Okay, so let's have a look at glycogen. So glycogen is another polysaccharide of alpha glucose, and very similar to amylopectin in terms of branching. Now, but the difference between amylopectin and glycogen is that glycogen has more branches, which means stored glucose can be released a lot more quickly. As I said before, if you have lots of branches, then enzymes can act upon it quicker because there's a larger surface area, break the bonds, and release energy a lot quicker. And this is obviously important for energy release in animals. It is also a compact molecule, so it is good for storage. The liver stores a large reserve of glycogen and continually breaks it down to maintain a stable glucose concentration. And you'll learn a bit more about glucose concentration in year 2. And we need glucose obviously for respiration. So can you describe the relationship of structure to function of glycogen? Knowing some bits from starch, it will be similar to glycogen. So the first three, it's too big obviously, so it can't leave the cell at all, or the cell surface membrane. It's insoluble, so therefore has no effect on water potential of the gut. So when you're talking about glycogen, we know that it's referring to humans, animals, so we have to mention the word gut. It's highly branched, so therefore an instant supply of glucose for respiration, therefore more energy is supplied. You could also say that because it's highly branched, it therefore can be quickly hydrolyzed, using a keyword here, for an instant glucose supply. So we've come across one type of bond so far, so that's a 1,4 glycosidic bond. Now the 1,4, as we know, is referring to the carbon number, but we also have another type of glycosidic bond called 1,6 glycosidic bond. You may have noticed this in the previous diagram, but both starch, so amylopectin, and glycogen have 1,4 and 1,6 glycosidic bonds. So let's have a quick look. So as you can see here, it's got alpha 1,4 glycosidic bonds, so that's just the normal straight chain. But then here we've also got another branch coming off here, and that's the 1,6 glycosidic bond. and so that's carbon number 1 and carbon number 6 trying to bond together. So looking at the previous diagrams, you can try and draw it out and imagine it. So let's look at cellulose. So obviously you should know that cellulose is found in plants. So cellulose is formed by the condensation of beta glucose. So all that time we were looking at alpha glucose, now we're looking at the polymer or polysaccharide of beta glucose. Now just to go over again the differences between beta and alpha glucose, it's the far right hand side, and make sure you know a way of memorizing the structure so you don't get confused between these two. So cellulose, this is the main substance in a plant cell wall. Like starch, cellulose is also a polysaccharide, but only made up of beta glucose. That is very important, because we don't want to say alpha glucose, we will just completely lose the mark. And each adjacent beta glucose is turned upside down, so I'll show you that in the next slide, and is joined by also 1,4 glycosidic bonds. And yes, you do need to know specifically that this is 1,4 glycosidic bonds, as it's also mentioned on certain mark schemes. Alternate bonding makes them straight, so as we saw before, there wasn't really alternate bonding in amylose. As a result, it was all coiled up, but here they're all straight, and that's because it's inverted each time. Each adjacent monosaccharide, beta glucose, is inverted, and each chain, as we can see here, is linked by weak hydrogen bonds. So if you're a chemist or taking chemistry A-level, you should know what this is. And collectively, these polysaccharides make microfibrils. So if you have lots of these polysaccharides together, so all these beta glucose chains, sooner or later they come together, lots of them, like strands or string or strands of hair, whatever you want to call it, they come together, and so many of them, and then they soon form what's called a microfibril. And this is to provide strength and structure to the cell wall in the plant, so it prevents it from exploding, and it just maintains its structure when there's lots of water inside, so it becomes turgid. So here, let me show you what I mean by inverted. So we know what a beta glucose molecule looks like. So we've got this group, beta glucose molecule here, we've got our glycosidic bond, 1,4, and you can see that this group, if it wasn't inverted, this group should also be here as well, but it's not, it's right at the bottom, so it's been inverted, and that happens constantly, and as a result we get straight chains. So you need to know the difference, or work out the difference between a reducing sugar and a non-reducing sugar. Annoyingly, teachers won't tell you what is a reducing sugar, what is a non-reducing sugar, so I'd be annoying and ask your teacher or A-level biology teacher what is a reducing sugar, what is a non-reducing sugar. So, but anyways, in A-level biology you don't really get asked what is a reducing, what is a non-reducing sugar, but because it's mentioned on the specification, I'd really make sure you know what is a reducing sugar. And if I explain it, it'll be overly complicated and you would want to die, so no, I'm not going to do that. So reducing sugars, examples that you need to know are reducing sugars, that's the main thing. You forget about knowing what a reducing sugar is, you need to know the examples. So you need to know that glucose is a reducing sugar, fructose is, lactose is, maltose is, and galactose is. We know that these three are all disaccharides, and these two are all monosaccharides. But sucrose, as we know, is sorry, my mistake. So as we know that glucose and fructose and galactose are both monosaccharides, and then lactose and maltose and sucrose are both disaccharides. Now, the reason why sucrose is all over here, I have no idea why. It's just a non-reducing sugar, and yeah, that's why why this is so special. It's just a non-reducing sugar, so it will have a different test for it. So we will be looking at how to test for reducing sugars, so these guys here, and non-reducing. So we're just going to look at how to test for sucrose essentially. So here is the steps on how to test for a reducing sugar. So we get this thing called Benedict's solution. Now Benedict's solution is also referred to as copper 2 sulfate. It's a blue substance. So first, get a test tube. Number 2, add your sugar in. So sometimes we can use this to test for sugar in urine, so we could also have our urine sample. No, don't do that. Then what you do is get your sugar solution, or the solution that you think has sugar in, but you want to see whether it does. So you add a Benedict's solution or copper 2 sulfate. Beware that they could just shove in copper 2 sulfate into the exam and say, oh, this Jonathan is going to add copper 2 sulfate into his test tube, blah blah blah. You need to know immediately that copper 2 sulfate is also called Benedict's solution. Number 4, heat gently in a water bath. So you get the water baths at school and you just insert your test tube into the water bath while it heats up. Number 5, observe for a color change. You now have made insoluble copper 1 oxide, which is a brick red color. And as a result, because it's a solid, insoluble, it will form a solid precipitate. And you do need to know the color change. And the color change is actually quite important. So blue indicates no reducing sugars, or it could also indicate a non-reducing sugar, but we don't know that yet until further tests. But for now, we know it's confirmed that blue means no reducing sugars. And the more the color change corresponds, so the more you have a color change, that corresponds to the strength or the concentration of the reducing sugar. So if we stay at blue, we know there's no reducing sugar. If we get green, then that means, okay, we've got some reducing sugar, but it's in weak concentration. Yellow is getting stronger, brown is getting even more strong, and brick red, ding ding ding, you've got really high concentrations of the reducing sugar. So if we compared a reducing sugar which was green to one that was yellow, immediately we know that the yellow colored sugar solution has a higher concentration. Sometimes we may have tested 5 different test tubes which all turn out to be brick red. So how do we know which one has the highest concentration? Was good you asked. So remember, the more color changes there are, the more precipitate is formed. Now we can use this concept of precipitate. So we use a technique called colorimetry, not calorimetry, colorimetry, so colorimetry, where we get some known standard concentration. So we get something that we know the concentration of. It could be one of our previous solutions we know the concentration of, and form a calibration curve. And then after forming the calibration curve, we can then find the absorbance, so how much light is absorbed from each sugar, and use the calibration curve to work out which sugar has the highest concentration. So basically we're getting a known value, plotting the known values, so we get sort of a calibration curve, and then so we can plot a graph of absorbance against sugar. So we can be in whatever unit, usually concentration of moles per decimeter cube. So we've got our sugar concentrations. We don't know what the sugar concentration is, so we can look at the absorbance, how much light it absorbs. So let's say it's right at the top here. We draw a line until it hits the curve, then we just drop down, and that should tell us the sugar concentration, or how strong the sugar is, and how much reducing sugar it contains. With the precipitate made, you could also centrifuge it, so put it into this spinning machine, and so that it all sinks to the bottom, so the solid, and you can then weigh each one, and obviously the heaviest one is probably the most concentrated one, or concentrated reducing sugar. Now we're going to test for non-reducing sugars, or the disaccharide sucrose. Now what we're going to do is add hydrochloric acid to the test tube of the solution. So let's say we've gone right from the beginning, so we've tested it and we've still remained at a blue, so that suggests that there's no reducing sugar present. So then what we do, we add heat a new sample with hydrochloric acid. Then so we've added hydrochloric acid, so the bonds of the sugar, because we know it's a disaccharide, we can break the bonds. So we can break this bond right here, and we know what sucrose is made of. So we know it's made up of glucose, and it's made up of glucose and fructose. And as we know that these two are both reducing sugars, and therefore it can go through this test as normal again. So I'm going to add hydrochloric acid to break the bonds of the sucrose, so we get the two monosaccharides which are reducing sugars, and then add sodium hydrogen carbonate to neutralize it. So this is an acid plus alkali reaction. Hopefully you remember that from GCSEs. So it results in salt plus water, and then add Benedict's copper 2 sulfate solution, Benedict solution or copper 2 sulfate solution, both the same thing. And then you're going to heat gently and observe for the same color change as we did before. No color change means that the solution is not a sugar and some other biological molecule and or compound or substance or whatever it is. And if there is a color change, then it is a non-reducing sugar. And usually whenever we talk about non-reducing sugars, we are referring to sucrose. So use a pH litmus paper after adding sodium hydrogen carbonate, as Benedict's solution will not work in acidic conditions. So that is really important. Whether you can add that in the exam if you want in your answer, it will probably make you look good. And plus, if you do this in class, then stick your hand up, ask the teacher, oh, did we use pH litmus paper, look all fancy, etc. But yeah, hopefully that should make sense. Okay, so now let's look at the test for iodine, potassium, the test for starch. So to test for starch, we add iodine to potassium iodide, then add your starch suspension in the test tube, then stir and observe for a color change from brown, orange color to a dark bluish black color. And yes, you could interpret this results blah blah blah. So you should know immediately every time you see a dark bluish black color, you know it tests positively for starch. That's it, and for the next lesson we will cover lipids. And hopefully if you've got any other questions or anything improvements or anything you want me to cover, please comment down below.